Hello again, students. Welcome to part one of the introductory microbiology slideshow. The purpose of part one is simply to introduce you to the subject of microbiology and also to the subdisciplines of microbiology. As I said earlier, biology is divided into two broad categories. One is studying living things that are big enough that you don't need a microscope to see them, and that's just regular biology. And then we have microbiology, which is the study of organisms that are small enough that you need a microscope to see them. So that would be microbiology. And as if that wasn't complicated enough, we divide the field of microbiology into several other disciplines as well, all of which are fun. I spent a certain period of my life studying microbes, and I honestly wish I'd spent more time doing that. But we need to learn the different subdisciplines first. And just some of the kind of career options that are available for people who have degrees in microbiology. We'll discuss that as well. All right, so traditionally, microbiology is divided into three subheadings. One is clinical microbiology. Clinical microbiology is the study of microbes that make you sick. So that is medical microbiology is another term for that. But there are also microbiologists who study microbes in the environment, microbial ecology, for instance. So the bad microbes that make you sick, they give all the other ones a good name, but a bad name. But in fact, only about 5% of the species of bacteria and other microbes that are found on the earth are actually bad for us. About 95% of them are actually good for us. They work to purify our atmosphere, to recycle things. Microbes, if you remember your ecology from Biology 110, microbes are some of the main detritivores that destroy, break down dead foliage on the forest floor. So when the leaves fall off the trees, they don't rot spontaneously. They are actually consumed by fungi and bacteria. There are other things. Microbes are decomposing our sewage and converting it into methane and things like that. So most of the microbes in the world, most of the microscopic organisms are either neutral to us or they're actually good for us. There are only about 5% of these species of bacteria and fungi and other things that are bad for us, and they unfortunately give everyone else a bad name. But if you look at universities around the world, you'll see that each university has a big section of microbiologists who are simply studying the microbes that are in the environment, and they're doing good things for us, and they're leaving us alone, or at least leaving us alone, or they're doing good things. Okay, so that's another branch of microbiology, environmental microbiology and microbial ecology. All right, the third main discipline of microbiology is industrial microbiology. This is people who use microbes to do useful things for industry. Okay, the simplest example, which I'm sure you've heard of, is alcoholic beverages, for instance. Alcoholic beverages are made with the help of microbes. So beer and wine, for instance, are made with the help of a fungus, a yeast called Saccharomyces cerevisiae. Bread is baked. Bread, you start out with dough, so you take flour and water and sugar and you mix it together. If you bake it, you'll just get a very hard mass. But if you want to have that nice soft bread with the pockets, the air pockets in it, you have to put in a bit of Saccharomyces cerevisiae, because Saccharomyces cerevisiae will start consuming the sugar and then generating carbon dioxide gas as a waste product, and that's what makes the bubbles in the bread. So the simplest example of industrial microbiology is brewing technology, or fermentation, as they call it. So when these microbes consume sugars, they consume polysaccharides, and they generate other products. That's an example of industrial microbiology. There are certain microbes that are used to treat fabrics. Sometimes you take wool and you treat it with cellulase enzymes that are produced by microbes, and then you make finer materials and so on. So there are lots of examples of industrial microbiology as well. This course has a strong bias towards clinical microbiology. I intended this course to be used by students who are intending to go into health studies or allied health professions. They can become doctors or nurses or other people who are interested in medicine or medically related fields. And so a lot of this course is devoted to talking about microbes that make us sick. However, don't think that all microbes are bad for us, because in fact most of them are good for us. Okay, so clinical microbiology is somebody gets sick, you assume, or the doctor or whoever assumes or suspects that it's the result of a microscopic organism. First thing you have to do is find out which microbe is making you sick so that you can find a specific way to kill it and make you better. Right? So that's clinical, that is, in a nutshell, that's the purpose of clinical microbiology. Okay, so one of the ways that you can do that is you can take a case history of somebody and you can say, what have you been doing? Have you been traveling recently? If you've traveled recently, where have you been? Certain parts of the world have certain microbes that make you sick, and we say that if those microbes are found only in that part of the world, we say that they are endemic to that part of the world, right? So there's certain parts of the world where certain microbial diseases are endemic, meaning that you commonly find them there. In British Columbia, we don't have a lot because microbial diseases tend to thrive in warm climates. So that means that most of the microbes that you find in the world, including the bad microbes, are found in the tropics because it's warm and moist in the tropical areas, right? So the tropical areas are near the equator and then a few degrees north and south of the equator where they get lots of sunlight. And so for that reason, during certain periods of history, they actually referred to the study of clinical microbiology as tropical medicine, because in the tropics, a lot of things made you sick, and they were usually microbes that lived in the tropics. So in British Columbia, we're a little bit lucky because we're farther north. We are not obviously in the tropics. It can be fairly cold in British Columbia. So there are fewer microbes around in British Columbia that make us sick, but there are a few. And for example, there is a disease called Lyme disease, which is endemic to British Columbia, and we'll talk about that later. It's caused by a bacteria called Borrelia burgdorferi. And there's another disease called rickettsial fever, Rickettsia rickettsii, which is also endemic to British Columbia. Also known as Rocky Mountain spotted fever because you find this disease in the Rocky Mountains. In BC, we have the Rocky Mountains in BC. Rocky Mountain spotted fever is more common if you go farther south. So if you're on the Rocky Mountains in British Columbia, you might catch rickettsial fever if you're bitten by a type of arthropod called a tick. The tick actually carrying the disease. But you're more likely to catch rickettsial fever if you're in California, for instance. Okay, so if you've been traveling, maybe you went to a tropical place and you had a drink. You know that there's certain tropical places where it's not safe to drink the water because the water is contaminated with all kinds of different microbes. And so you go to these tourist resorts and you order a drink, and they filter the water that they put into the drink to make sure that you don't catch these diseases. However, a lot of times these resorts don't filter the water that they freeze to make ice cubes. So the ice cubes in your drink may melt and then release their many pathogenic microbes that are bad for you that can survive being frozen, and you get sick. So these creatures are called Giardia lamblia, which is a protozoan, and they can make you sick. They can survive being frozen in an ice cube. There are other organisms called Entamoeba histolytica, which causes a disease called amoebic dysentery. Amoebic dysentery is a violent, bloody diarrhea, which causes severe dehydration, and it's potentially lethal. Potentially lethal means it could kill you. So finding out what makes somebody sick, part of that, the easy part, is just to say, what have you been doing? Where have you been? And then if you're a doctor who's trained in that sort of thing, that can give you clues about what it might be. Okay, then once you have clues about what it might be, you take samples of bodily fluids. You take samples of blood, you take samples of mucus, you take samples of sputum, which is the spit, basically. Sputum is a fancy word meaning spit or saliva. And then you look in there to see if there are any microbes in there. Okay, so one of the most common tools that microbiologists use is this thing here, which is called a Petri plate. This is a Petri plate. And what you do, this thing here that looks like a little stick, that's called a sterile loop. So at the end of this little stick, there's a wire loop that you sterilize by heating it up, by putting it into a flame. And then you dip that loop into the patient's blood or into the saliva or the sputum or the stool. The stool is a fancy word for excrement. And you then streak it on one of these Petri plates, which is filled with food. This is a specific type of Petri plate that contains a type of food called agar. Specifically, it's called blood agar because they mix sheep's blood with it. And then if there are any bacteria that are on the end of that loop, you put the Petri plate into an incubator, and those bacterial cells will grow into a colony, a large colony. So these big puffy round things that you see on the Petri plate are individual colonies. Theoretically, each one of those colonies arose from an individual bacterial cell. So if you put one cell onto a Petri plate and then you put it into the incubator, that cell will divide in two in a few minutes. It'll divide into 2, and then into 4, and then to 8, and then into 16, then 32, 64, and then it doubles every 20 minutes or so. If you leave it in the incubator at a nice warm temperature, after 8 hours, it will actually grow into a big lump of bacteria, a big pile of bacteria on that plate, which is called a colony. And then from that colony, you can then take a sample of that colony and you can look at it under a microscope, and you can look to see whether the bacteria are rod-shaped or round, whether they are bacillus or cocci, for instance. And then you identify them by morphology. So by looking at the shape and size of the colonies, that's one example of morphology that you can use to identify which bacteria is making you sick, for instance. But also, if you look at the bacteria themselves, you can judge which bacteria it might be based on the morphology of the actual bacterial cells. So here we're looking at the morphology of the colonies. And then here we look under a microscope and we look at the morphology of the actual bacterial cells themselves. Okay, so you see that these cells are longer than they are wide, so these are bacilli. And then notice that they have these little whips that are coming off them that look like hairs. Those are flagella that they use to swim. And this particular bacteria is completely covered with flagella, and we have a term for that in microbiology. You call that paratricious. A paratricious bacterium is a bacteria that's completely covered in flagella. We have names for other bacteria that only have a flagella at one end or have one flagella at each end. Right? So those are, again, examples of identification of bacteria based on morphology. Okay, so if you're a technician in a microbiology lab, you spend a lot of your day, or you may very well spend your entire day, doing something called streak plating. That's where you take samples of blood like we have here in these test tube racks, right? And then we have a stack of plates, and then we streak the loop onto the bacterial plate and put them in the incubator and come back the next day and see what grows. And so that's what microbiology technicians do. Being a technician is usually a job that's done by somebody who has just a bachelor's degree in microbiology, and it's not a very pleasant job. It's fairly tedious, but you're working with bacteria. It is a job. It's relatively well paying if you work in a hospital. In order to work in a hospital, you have to be a registered medical laboratory technician. You have to have a certificate called an MLT, which you can get in 2 years. So you would typically go to Columbia College for 2 years, and then you go to BCIT and you get an MLT certificate. The MLT certificate allows you to work in the hospital laboratory doing not just microbiology tests, but doing a dozen types of other tests. So if you're an MLT who works in a hospital laboratory, the job is not quite so boring because they try to rotate the jobs. So you spend some of your time streak plating and making plates, and you spend some of your time doing polymerase chain reactions, and you spend some of your time doing other things. So, but anyway, if you work in a laboratory that does nothing but microbiology, you could very well spend all of your time just doing this, streaking bacteria onto plates. As I said, one of the other things you could do as a medical laboratory technician is to do the polymerase chain reaction. The polymerase chain reaction, this is a thermal cycler, a PCR thermal cycler. You might have heard in other courses that I've taught that this polymerase chain reaction is a way of rapidly increasing. Let's say that you have a gene that's present, a piece of DNA that contains a gene that is unique to a certain bacteria. In order to identify that gene as being specific to a bacteria, you have to make many, many copies of it for you to analyze. And the quickest way to do that is by doing a polymerase chain reaction. You'll learn a lot more about the polymerase chain reaction in either Biology 200 or Biology 234. But for now, in this course, let's just say that PCR is a way of making multiple copies of genes. And if you can make multiple copies of genes, it's a way of identifying whether or not bacterial genes or specific bacterial genes or specific viral genes or specific yeast or fungal genes are present. And so, if you suspect a person is being made sick because they've been infected with a bacteria called Staphylococcus aureus, you can take a sample of their blood, and then you can do a PCR reaction to see if there are Staphylococcus aureus genes in their blood. Right? So it is in the context of clinical microbiology, a PCR test can be used as an identification tool to identify what it is that's making somebody sick. Okay, once you've determined what makes somebody sick, you have to identify something that can kill it. These things in the bottles over here are known as antibiotics. You might have been given an antibiotic sometime. What you may not have known is that some antibiotics will kill this type of bacteria, but not that type of bacteria. Right? So in order to do a good job of killing the bacteria, you need to know exactly which bacteria it is, if a bacteria is making you sick. If you're being made sick by a virus, there's another set of things you can do. If you're being made sick by a helminth or a protozoa, there's another set of things you can do. But if you're being made sick by a specific bacteria, you can be cured with a specific antibiotic. There are things that are called broad spectrum antibiotics that will kill a number of different bacteria, but those broad spectrum antibiotics tend to be hard on you. They tend to damage the person that the bacteria is in, whereas a specific antibiotic that will only kill one specific bacteria tends to be less hard on your body. So typically if somebody is sick with a bacteria, if it's not a serious infection, they'll wait a day or two to do some tests to figure out which bacteria is making them sick so that they can prescribe a specific antibiotic to kill it. On the other hand, if they're very sick and they need immediate treatment, and you don't have time to figure out which bacteria is making them sick, then you would give them a broad spectrum antibiotic for a couple of days until you do the tests to figure out which specific bacterium is making them sick. All right, now clinical microbiology is traditionally divided into 4 different subcategories. So we've already divided microbiology up into clinical microbiology, microbial ecology, and industrial microbiology. Now we're going to divide clinical microbiology even more. Okay, so bacteriology is the study of bacteria. That is a branch of clinical microbiology. The term mycology is specifically the study of fungi that make you sick, right? So bacteriology is the study of bacteria that make you sick. Mycology is the study of different fungi that make people sick. Right? And when you get a disease, when a fungus makes you sick, we call it a mycosis. And so there are several different types of fungal mycosis that you can become infected with, and we'll talk about what those are. Parasitology is the term given to the study of protozoa that make you sick, or helminths, or sometimes the insects that transmit the parasites back and forth between animals and humans, or between humans and humans. So parasitic diseases that are caused by worms or protozoa is generally referred to as under the umbrella title of parasitology, because these are parasites. Now, technically, the bacteria that are making you sick and the fungi that are making you sick are also technically parasites, but for some reason, just because of the way the science of microbiology has evolved, we call the study of protozoa and helminths parasitology. And as I said, sometimes parasitology is also sometimes referred to as tropical medicine, because you find a lot of these parasites in the tropics where it's warm and moist, whereas once you get into a colder climate or a drier climate, there are many fewer things that can, only a few things can grow in a cold climate, and only a few things can grow in a dry climate. Finally, the study of viruses that make you sick is specifically called virology, and so there are some microbiologists that spend their whole careers just studying viruses, and it is a totally different field of study that requires a totally different body of knowledge than bacteriology, for instance. So viruses have very different characteristics than bacteria. They can be killed or destroyed in ways that are very different from bacteria. So these are all specialties. Now, if you go to Vancouver General Hospital, if you might have seen where Vancouver General Hospital is in Vancouver, you can actually see the building from Columbia College. Next door to Vancouver General Hospital, there's a small, inconspicuous little building that is called the BC Centre for Disease Control. The BC Centre for Disease Control is the government agency that is responsible for keeping track of microbial diseases, communicable diseases in Vancouver, in British Columbia rather. Right? So whenever somebody gets diagnosed with a specific type of a disease that is caused by a microbe like a protozoa or a virus, the doctors in the hospitals have to report these statistics to the BC Centre for Disease Control. And the BC Centre for Disease Control is filled with microbiologists who are studying infectious diseases that are caused by microbes. And interestingly, the BC Centre for Disease Control, the building itself is divided into four different floors that have these specialties. Right? So on the ground floor, on the bottom floor, is the bacteriology division, and the people on the ground floor are studying nothing but bacteria. Then if you take the elevator up one floor, then you go to the mycology section where everyone is studying fungus. If you go to the third floor, everyone is studying malaria, parasites, and Lyme disease, Borrelia burgdorferi. People are looking at microscopic ticks under the microscope. And then if you go to the top floor, you won't actually see any incubators or anything like that, because viruses don't grow in those conditions. But the people at the top are studying just viruses. And so they do this, they divide the building up this way specifically to avoid cross-contamination, because if you had one laboratory that was studying fungus, and then next door to it you had another laboratory that was studying bacteria, it's not uncommon for some fungal spores to float over through the air and contaminate the bacterial cultures in the lab next door, which they don't want. So typically, institutions that study microbiology this way are physically divided into different sections of the building, so that the different types of microbes don't contaminate each other. All right, now let's just briefly discuss environmental microbiology and ecology. As I said, only about 5% of the bacteria and fungi are actually bad for us. We end up hating all of them because they get a bad name. But please do not hate the bacteria or the fungi, because 95% of them are actually doing a wonderful job recycling our waste products, allowing, for example, bacteria. For example, you know that there's nitrogen gas in the atmosphere, and you know that when plants grow, they turn the nitrogen gas into amino acids. Amino acids are necessary to make proteins. Well, as it happens, there are many plants that we rely on for food that cannot convert nitrogen gas into amino acids. That job is done for them by symbiotic bacteria that are able to do something called nitrogen fixation. So there are many plants. Most of the beans and the legumes that we eat for food live in a symbiotic relationship with nitrogen-fixing bacteria. So without bacteria, there about half the things that human beings rely on for food would not exist, because those things, those plants don't have the ability to fix nitrogen gas and convert it into amino acids. So, as you know, as you recall from Biology 110, hopefully, a symbiotic relationship is where two organisms live close together and they benefit from each other's interaction. They live close together, they interact with each other, and they both benefit, both organisms benefit. Another type of interaction is called a parasitic interaction, and that's where two organisms live together and interact with each other, but only one organism benefits from the interaction, and the other organism is actually damaged by the first interaction. That is referred to as a parasitic relationship. Whereas these relationships that exist between plants and nitrogen-fixing bacteria is a classic example of a symbiotic relationship. The plant gets the bacteria converts nitrogen gas into ammonia, which the plants can then convert into amino acids. That's what the plants get out of it, and that's what the bacteria provide. Meanwhile, the plant provides a nice, safe place for the bacteria to live. The bacteria live inside the roots of these plants, inside what are called root nodules. Okay, so as I said, 95% of the microbial species are actually good for us. Only 5% give the other 95% a bad name, bad reputation. Now here's an interesting thing. The Earth, we believe the Earth is about 4.5 billion years old, and there was no life on Earth for the first 2 billion years. And then for the next billion years, there was nothing but bacteria. And then for the next billion years, there was nothing but bacteria and protists. Protists are single-celled eukaryotes. Remember that a eukaryote, of course, is an organism that keeps its genes in a nucleus, whereas a eukaryote keeps its genes in a nucleus, whereas a prokaryote does not have a nucleus. And in addition to that, a eukaryote is larger than a prokaryote. So bacteria are prokaryotes, for instance, whereas humans are an example of eukaryotes. In fact, humans are an example of a multicellular eukaryote that's made of billions of cells as opposed to just one cell. Now, somewhere in between the prokaryotes, like the bacteria, and the multicellular eukaryotes like humans, in between we have the protists, which are single-celled eukaryotes. Now, during the second billion years of life on Earth, the predominant life form was the protists, and they were all living in the ocean. Right? So for the first 2 billion years of life on Earth, the first billion years we just had bacteria and they were living in the ocean. The second billion years we had bacteria and protists that lived in the ocean. So for a billion years we had protozoa that were protists. Sorry, I'll explain the difference between protist and protozoa in a minute. But for a billion years we had protists in the ocean that were living and dying in the ocean. And where there were certain spots where lots and lots of protists lived, and then they would die and they would float down to the bottom of the ocean, and then the skeletons would the decomposing bodies of those protists, those microscopic protists, would be there, and then more protists would die and fall on top of them, and they would bury each other and then compress. They'd be compressed by the various layers that fell on top of them for hundreds and hundreds of millions of years. And this compression caused complicated hydrocarbons to form, cyclical hydrocarbons and things, which we now call crude oil. Right? So there is a specialty of environmental microbiology where you can specialize in identifying the specific types of protists that you would find on the ocean floor, and use that information to predict where oil is located underneath the ocean floor. Right? So people who explore and look for oil in the ocean, underneath the ocean, look for these things. And these are they look for these things on the ocean floor, and these are protists that you find in the ocean. Right. So, in fact, one of the most highly paying jobs that you can get as a microbiologist, generally microbiologists, the environmental microbiologists are not very well paid. They tend to work at universities and they're studying uh, soil bacteria and things like that for the sake of understanding how they work. But one of the most highly paid jobs you can get as a microbiologist is a microbiologist who specializes in identification of prehistoric ocean protists. And so these drilling ships go around the ocean and they drill cores out of, they drill samples out of the ocean floor, and then they get the microbiologists to look at the soil, or at the rock that they've drilled out of the ocean floor, and then you say, oh well, look, I see a whole bunch of these things. I see a whole bunch of these protists. Probably there's a rich deposit of oil just underneath them, so that's where you should look for oil. So there are some microbiologists that actually work in the petroleum industry. They work they have jobs identifying areas of the ocean floor under which there is probably a deposit of oil. On to industrial microbiology. Fermentation is the conversion of sugar to alcohol. One of the most common types is where you take grapes, which are filled with sugar. They're filled with sucrose and glucose, and you crush them up and then you put them into a bottle and you leave them for a while. And on the outside of those grapes, there was a yeast called Saccharomyces cerevisiae, as well as a dozen different types of bacteria. Right? And those bacteria, and especially the yeast, will ferment the sugars into alcohols. Right? Specifically, Saccharomyces cerevisiae is a very common yeast that you find everywhere. And it just so happens that Saccharomyces cerevisiae, when it encounters glucose, if it encounters sucrose, it breaks it into glucose and galactose, glucose and fructose, and it digests the glucose and converts it into ethanol. And so for centuries, for thousands of years, in fact, humans have taken fruits and vegetables and crushed them up, and then noticed that if you leave it long enough, it turns into an alcoholic beverage. The reason for that is because those vegetables or those fruits had Saccharomyces cerevisiae sitting on top of them, on the skin, on the outside of the fruit, and then when you crush them up, they started getting to work digesting the glucose and converting it into ethanol. Okay, so one of the most common uses of microbes like Saccharomyces cerevisiae and other types of not E. coli, but other types of bacteria. Saccharomyces cerevisiae is a yeast. The most common use is fermentation of alcoholic beverages, including wine. All right, now this is interesting. Making wine is not actually a haphazard art. I mean, you've probably heard of the way they used to do it in the old days. They'd throw all the grapes into a barrel and then they'd crush them with something called a grape press. Or even worse, in Italy, the peasants used to get into the barrel with their bare feet and stomp the grapes with their bare feet, which is kind of disgusting to think about. And then you would filter out the grape skins and then just take the grape juice. And if you put that into bottles, it would ferment into wine. But in fact, these days, making wine is a highly sophisticated art form that involves a lot of science. And the science of making wine by studying the microbes that ferment the wine is called enology. Enology. And this is it's actually a very interesting science. And people who do this for a living, they very carefully control which types of microbes get put into the grape juice. They control the pH of the grape juice. They control the sucrose content and the glucose content and the fructose content. And if there's too much sucrose and not enough glucose, they will adjust that. If the pH is too low, they'll adjust that a little bit, and then they will deliberately seed or inoculate the grape juice with a certain fixed amount of Saccharomyces cerevisiae in order to get exactly the right flavors. So in the old days, we used to get the best wines from Bordeaux in France, or from Burgundy in France, because that's where they assumed that's where the best grapes were. In fact, that's really just where the best microbes were. Right? So as soon as somebody figured out that the best wines were being fermented by a certain percentage of Saccharomyces cerevisiae and Lactobacillus acidophilus, they could recreate that combination, and now all of the wine that we can buy in the store, more or less, it tastes more or less as good as the wine from Bordeaux or from Champagne or any place like that in France or Italy, for instance. Okay, now just to reinforce the idea that studying the things that ferment wine, grape juice into wine, is a science, UBC, which is the largest university in Western Canada, UBC actually has a wine research institute. It's called the UBC Wine Research Center. It's a big building on the UBC campus where they do nothing but analyze and study ways to make better wine. This is the head of the BC Wine Research Center, a famous professor named Henny Van Vuren. And so UBC has a big department that is devoted, or big research institution that is devoted exclusively to studying how to make better wine. The reason why would we care about this? Because winemaking is a big industry in British Columbia. If you go to British Columbia, the interior, Kelowna, Kelowna is covered with grape vineyards, and we don't have a long growing season in British Columbia. We don't get a lot of sun, but we have just enough sun to make certain types of wine. And so the people at the UBC Wine Research Center are studying what are the best microbes that you can put into the kind of grape juice that is produced in British Columbia to make the best tasting wines. And so British Columbia has a very large winemaking industry, and it would be nice if some of you went into enology and went to work at the UBC Wine Research Center and helped British Columbia grape, helped farmers to grow grapes in Kelowna, or in the Okanagan and Kelowna, and then use your knowledge to make the best types of wine for British Columbians. And wine is also one of the main things that British Columbia exports to the rest of the world. We don't make the best wine in the world, but it's not bad. You know, it's pretty good wine. It's as good as anywhere else. Okay, now if you take something else, if you take barley and you allow the barley seeds to germinate, and then you put Saccharomyces cerevisiae in with those, you make Saccharomyces cerevisiae, the yeast again. If you put Saccharomyces cerevisiae in with barley and some other fruit called a hops, you allow it to ferment, you will have made beer. Right? So they've known this. This is a kind of a Western European thing. Beer was always very big in Germany and England, and the French never drank beer, really. It wasn't their thing. They liked their wine, and the Italians liked their wine, but the Germans and the British and a few others liked their beer. And that is basically made by mixing Saccharomyces cerevisiae with barley and hops. And you put them in these large machines called fermenters, and these large machines, they generate a lot of heat, and they generate a lot of bubbles, and they generate a lot of excitement. And this is a very complicated science to brew good tasting beer, to ferment good tasting beer. And so this, if you drive over by the Granville Street Bridge, you'll see this big building over by the Granville Street Bridge, which is the Molson Brewery. There's the name Molson. Molson is one of the biggest, Canadians are very big on beer as well, so are Americans. And so one of the biggest brewing companies in Canada is the Molson Brewery, which you'll see over by the Granville Street Bridge, by the southern part of the Granville Street Bridge. And these giant tanks are filled with brewing beer, filled with water and sugar and hops and barley and Saccharomyces cerevisiae. And this is a complicated manufacturing plant, essentially. And the person who controls this whole process, a very big important job, is somebody who's called the brewmeister. Brewing, of course, is making these components into beer, and Meister is a German word meaning master. Right? So the brewmeister is the scientist who is in charge of controlling the production of beer, which is, again, it's not a cottage industry, it's a science. And so exactly how much Saccharomyces cerevisiae do you put in, and when, and how much sugar does the barley have, all of that is something that is controlled by the brewmeister in order to make a good tasting beer. So that's another thing that you can go into if you know about microbiology. Okay, Many of the enzymes that we use in industry, you use enzymes to digest wood pulp to make paper. You make enzymes that digest cellulose to make acid wash jeans and to make washing powders that take the stains out of jeans. Many of these enzymes that we use for industrial purposes are produced in bacteria. They can be isolated from other places, but if an enzyme is a protein catalyst. Uh, if you have the gene that codes for the enzyme, then you can make that protein, you can make that enzyme in Escherichia coli, for instance, which is a very common bacteria. Right? So there are many things that we use enzymes for in industry, and those enzymes are usually produced in bacteria as part of a recombinant DNA technology, where you take the gene for the enzyme out of the organism that makes the enzyme, and you put it into E. coli instead, Escherichia coli, and then you produce it in E. coli and purify it. Okay, here's another example of using microbiology for industrial purposes. There are many bacteria that you already know that Saccharomyces cerevisiae will convert sugar like glucose into ethanol, right? We know that. You also know that you can run cars if you mix. You can run ethanol is flammable, right? So anything that burns can be used to power steam engines or even car engines. Right? So ethanol is flammable; it burns. So one of the things that people do is they make something called gasohol. Gasohol, of course, is the combination of the word alcohol and gas, gasoline. And so what they do is they take ethanol that's been made through fermentation, and they mix it with gasoline at a ratio of about 85% gasoline, 15% methanol, and they refer to that as gasohol. Gasohol, and you can put that in the car's gasoline tank. Okay, so this is a way of getting away from fossil fuels. So normally gasoline for automobiles is made by taking crude oil and then refining it until you have petrol, until you have gasoline. We're running out of fossil fuels, and so one of the alternatives is to use ethanol to basically stretch the gasoline so that the gasoline that we have or the oil that we have will last longer if we supplement it with ethanol as well. Right, so that's called gasohol. Now, normally sugars are hard to get, right? And any crops, any food that we grow, any plants that we grow that contain large amounts of sugar, we'd rather use that for food. But there are some plants that, some parts of a plant that we can't eat. And so wouldn't it be nice to turn those parts of those plants that we can't eat, turn those into ethanol and then use it to make gasohol? Best example is corn, right? So you know that the corn kernels we can eat. So humans and other animals can eat the corn kernels, but the stalk, the stem of the corn, we cannot eat that. So there are a lot of scientists who are devoting energy to finding bacteria. There are some bacteria that can eat it. Those stems are made primarily of cellulose. Cellulose, of course, is made of beta glucose, and humans don't have the digestive enzymes to digest beta glucose. However, there are many types of bacteria that produce enzymes called cellulase enzymes that can break down beta glucose and convert it into alpha glucose, that can break down cellulose into beta glucose and then basically change the structure into alpha glucose and then ferment it. So there's a large body of scientists who are working on converting the stems, the stalks of wheat, and the stalks of, the stalk of wheat is called straw, and then also converting the stems or the stalks of corn plants into alcohol using cellulase enzymes that are produced by bacteria. Now, there are some bacteria that can actually take human excrement or animal excrement, and when humans eat food, we can't digest all of it, right? So we know that some of it comes out in a kind of an unpleasant, smelly substance, human excrement. The same thing happens with animals. And when it comes out of humans or animals, we've digested everything we can digest from that food. We've gotten as much energy as we can get out of that food. But there are some bacteria that are more than willing to keep on digesting it, and will keep on digesting the things that we couldn't digest. And generally, those bacteria digest, those are more primitive types of bacteria that digest sugars and proteins and things that are in excrement and convert them into methane gas. Right? Methane gas. And methane gas can then be converted into methanol, the alcohol version of methane gas, methanol. And you can also mix the methanol with gasoline, with petrol, in order to make something called biodiesel. So there is a large body of scientists who are working on cultivating bacteria that are good at converting sewage, converting human sewage and animal waste and animal excrement into methanol. First, it's converted into methane gas, and then you convert the methane gas into a liquid. You make methanol, and then you mix that again. You mix it with gasoline at a ratio of about 85% gasoline, 15% methanol. All right, now this is not a very pleasant subject, but these are hog farms. You know that many of you don't eat pork. I congratulate you for not eating it because it's not very good for you, and it's also, you know, I'm on principle I'm against eating animals, but I do anyway. Like many North Americans, I'm a bit of a hypocrite for when it comes to eating animals. But if you eat pork, the pork has to come from somewhere. It comes from large farms these days that are called hog farms. And the pigs eat a lot of food and they produce a lot of excrement. And the farmers that grow pigs to eat have trouble finding ways to get rid of the animal excrement. And so here, this is a large hog farm in Arkansas, a state in the United States. And this thing here that looks like a square lake is called a manure lagoon. And it is filled with pig excrement. And it reeks because there are thousands and thousands of liters of pig sewage in that lagoon. And they have trouble getting rid of this, right? It seeps into the groundwater, and the pig sewage contaminates the groundwater. So over here, you can see in the distance, there's another, these little houses are more houses where the pigs live, and there's another manure lagoon over there. And you can smell these lagoons. You can smell them. They reek, and you can smell them for miles. And the excrement gets into the groundwater and contaminates it, and they don't know what to do with it. Well, that's actually quite foolish because there's a bacteria called Methanopyrus candeleri that will convert all of that pig excrement into methanol, into methane, which can then either be used to power things with methane gas, or you can convert it to methanol and use it to run automobiles. And so there are some farms in the world where they use this method to generate methane gas. This is particularly useful in the third world, where you have rural parts of underdeveloped countries, rural areas where they don't have electricity, but they do have animals. So you take all of the animal sewage, the animal excrement, and you put them into tanks with these bacteria. The bacteria will convert the sewage into methane, and then you can use the methane to run a generator, for instance, to power electricity, to make electricity, for example. So there are many farms around the world that are now using the methane generated by animal waste to create electricity, or to do other things, to generate heat to cook food, or to make steam engines, or even to convert into methanol to mix with gasoline and make biodiesel. All right, another use for bacteria and other microbes is in pharmaceuticals. So as I said earlier, when we were talking about enzymes, enzymes can be made by all sorts of animals, but generally if we need enzymes for some industrial purpose, we tend to just take the gene that codes for the enzyme and put it into bacteria, and then produce the enzyme in bacteria because it's easier to isolate the enzyme from the bacteria. Those of you who took Biology 200 know that the easiest way to purify a protein that you've produced in bacteria is simply to take the gene and engineer it so that you put 6 histidine amino acids onto one end of the protein. You would then call that a recombinant protein because you've recombined the genes. Right, so you take the gene and you make a recombinant gene, you put 6 histidines onto the end of the protein, and then all you have to do is break up the bacteria, and then you pass the homogenized bacteria through a column called a metal chelation affinity column that purifies the protein right away. So many of the many of the proteins that we use for enzymes in industry are produced in bacteria. And furthermore, there are many proteins that we use for pharmaceutical purposes, for drug therapy, that are produced in bacteria. All right, so as good Canadians, you should know who this guy is. This is Frederick Banting, who is a famous Canadian scientist. He was famous for discovering insulin in 1921. And in 1923, he was given the Nobel Prize for that discovery, and it was the first Canada's first Nobel Prize. Banting originally discovered insulin in the pancreases of dogs, and then eventually, so it was used. Insulin is used as a therapy for diabetics. So diabetes used to be a disease that was lethal. It was deadly. You wouldn't live to be in your 20s. You would die as a teenager or in your early 20s from diabetes. But now you can live your entire life as a diabetic if you inject insulin. Okay, so Frederick Banting won the Nobel Prize in 1923 for discovering discovering insulin and then how to use it and make it. Now, initially, the insulin was produced. He discovered it in dog pancreases. Then they said, okay, well, where are we going to get enough insulin? Where are we going to get pancreases in large enough quantities to make enough insulin for all the diabetics? Well, they initially got the pancreases from all the pigs that were being slaughtered for food. Right? So here we have a metric ton of pig pancreases, and after you use the chemical process to extract the insulin from these pancreases, you end up with 1 liter of insulin, which is shown in the foreground here. Right, so you have to take a ton of pig pancreases to make a liter of insulin. That's not very efficient. And furthermore, there may be some people who don't like the idea of being injected with a pig protein, right? Putting something from a pig's body into your own body. Right? So what they did was they simply took the gene for insulin out of a pig, and then they put it into a bacteria. They put it into a special thing called a plasmid. Right, so a small circular piece of DNA called a plasmid, you take the gene for insulin out of the pig, you splice it into a bacterial plasmid, you put the bacterial plasmid into E. coli, into Escherichia coli, which is a bacteria, and then the bacteria will produce the insulin for you, and then you can purify it. Right? So they used to produce insulin by grinding up a whole ton of pig pancreases and injecting a pig protein into people, right? And so some people didn't like that idea, what if, just at the simplest form, what if you're Jewish or what if you're Muslim, right? Then you don't like the idea of pig tissue being injected into your body, definitely, right? So in this case, what they did was initially they took the pig gene and they put it into bacteria, and then they produced protein in bacteria. Now, you might ask the question, does that, you put a pig gene into bacteria, does that mean that the bacteria is now producing a pig protein? Well, at the time they did this in the 1970s, most scientists were of the opinion that no, that's not a pig protein anymore. As soon as you move the pig gene into bacteria, it's not really a pig protein anymore. Yeah, the gene came from a pig, but the protein itself is being produced in bacteria. So a lot of people didn't have a problem with injecting the recombinant protein. However, there was a Saudi prince who was a Muslim who had a son who was a diabetic, and he didn't like the idea of his son being injected with a protein that was produced in bacteria from a pig gene. So he donated some money to an American company to reclone, to do the genetic engineering over again with the human gene. So now the protein that diabetics inject into themselves is produced in E. coli, and it is encoded by a human gene, not an animal gene. Right? So anyway, that's just an illustration of how the pharmaceutical industry uses bacteria, which are microbes, in order to make many of the proteins that are used for pharmaceuticals. And the best example of that is insulin. So there are some pharmacies that have giant fermentation tanks like the ones that you see at the Molson Brewery, except that instead of making beer, they're busy making insulin from bacteria. Okay, so what are we going to learn during the rest of part one of this lecture? We're going to learn the difference between eukaryotes and prokaryotes. We're going to learn how to name various types of microbes using the binary nomenclature. This is something you should remember from first year, right? And you should know the difference between a prokaryote and a eukaryote from first year. This is just review, right? And then we're going to take an example. We're going to take a short tour of several different types of microbes, and that's what's going to happen in part two of this lecture. And then we'll have some practice questions which you can practice. And then after part two is over, we will go on to part three, which is a brief discussion of how microscopes work. So if you're studying microbiology, you're studying life forms that are so small you need a microscope to see them, and so that's what you're going to study in part three of this lecture. All right, let's do a brief review of the difference between a eukaryote and a prokaryote. Okay, as I mentioned earlier, prokaryotes have been around for 2 billion years longer than humans or any other animals. We believe that the world is 4.5 billion years old. For the first 2 billion years, there was no life at all. For the next billion years, the third billion years, there was just bacteria in the ocean. That's a long time where we just had bacteria. And then for the next billion years, in the fourth billion years, protozoa, protists, were evolved, and then so we had another billion years where we just had bacteria and protists, and they were in the ocean. And then it wasn't until the last 300 million years or so that things came out of the ocean, plants and so on, and then animals evolved in the ocean and came out of the ocean. All of that stuff happened just in the blink of an eye, as far as nature is concerned. It just happened in the last 350 or 400 million years, which is no time at all compared to the amount of time that bacteria have been around. All right, so bacteria, the main difference between a prokaryote and a eukaryote is that a prokaryote does not have a nucleus, right? So a prokaryote is another term, the fancy official term for a bacterium, right? So in addition to that, bacteria tend to be smaller, right? So a typical bacteria is 0.1 to 10 micrometers in size. Remember that a micrometer or a micron is the scale that microscopes or microbiologists like to work with. So if you take a meter and you divide it up into 100, you have centimeters. If you take a meter and you divide it up into 1,000 parts, you have millimeters. If you take a meter and divide it up into 1,000,000 parts, you have a micrometer. Right? So a micrometer is 1/1,000,000th of a meter. It's 10 to the minus sixth meters. Right? And typically a human cell or a typical eukaryotic cell is around 10 microns in size. Right? So you see the scale down here, 10 microns to 100 microns is the typical size for a eukaryotic cell. 0.1 microns to maybe 1 micron is a typical size for a bacteria. They list here 0.1 to 10 micrometers. Believe me, there are very few microbes, very few bacteria that are 10 micrometers in size. In fact, I don't even know of any offhand. So this scale on the picture is a little bit off. But typically, a prokaryote, a bacteria, is 1 or 2 microns in size, whereas at the most, whereas a eukaryotic cell is 10 microns or larger in size. And a eukaryotic cell has a nucleus where the DNA is stored. A eukaryotic cell also has linear chromosomes, so each chromosome is a single piece of DNA. Do you remember from first year what type of chromosomes bacteria have? They have circular chromosomes. Generally, a bacteria, a prokaryote, has a single circular chromosome. In addition to that, eukaryotes have membrane-bound organelles like mitochondria, and an endomembrane system like the rough endoplasmic reticulum and the smooth endoplasmic reticulum and the Golgi complex. Right? So bacteria prokaryotes do not have any of those things. Prokaryotes don't have mitochondria because they're not big enough for that. In fact, as you might remember from first year, we believe that the mitochondria inside eukaryotes actually evolved from bacteria. They used to be independent living, free living bacteria that have established a symbiotic relationship with eukaryotic cells. So at some point, 2 billion years ago, 1.5 billion years ago, a eukaryotic cell that didn't have mitochondria swallowed a bacteria, and instead of digesting it, it just kept it as an energy slave, basically, and then that persisted because these bacteria, these symbiotic bacteria that became the mitochondria, they produced so much spare energy that those eukaryotic cells that had mitochondria thrived and survived much better than the ones that didn't. But as you'll see in this course, we'll discuss some of the protists that do not have mitochondria because they evolved before this happened, right? And they're still around. All right. Taxonomy is a branch of biology where you determine the evolutionary relationship between things based on their structure, right? So similar structure, and then you can look, phylogeny is actually, taxonomy is where you put things into groups based on their structure. It's a branch of biology. And then phylogeny is where you put things together based on their structure and assume that they have a similar structure because they evolved from a similar ancestor. That's called phylogeny. Okay, so then how do you name all of these things that you've put together into different phyla? Well, an official naming system is referred to as a nomenclature. A nomenclature, official way of naming things. And in biology, we use what's known as binary nomenclature. Binary, of course, means two, one of two, and nomenclature means the official way of naming things. And the way we do it is every organism belongs to a genus, and every organism has a species. Right? And then generally, every organism belongs to a domain, and we actually have three different domains that we worry about. One of them is the domain bacteria, right? So all of the prokaryotes belong to that domain. And then there's the domain eukarya, and every organism that has either, whether it's a single-celled organism or whether it's a multicellular organism, belongs to the domain eukarya. And then within those two domains, there's another domain called archaea, which we'll talk about later. But within those domains, you have different phyla. So phyla are like giant lineages. And then within phyla, you have several families. Within families, you have several classes. Within the classes, you have several genii. And then within each genus, you will have several different species. You remember that. Now, the interesting thing is that you remember from first year biology that a species is defined as the ability to mate and produce viable offspring. The word viable means that they'll survive and thrive, right? And so how do you define two things being members of different species? We define them as being members of different species because they can't reproduce and produce viable offsprings. Okay, that is the traditional way to define a species. Right? Now, we run into a little bit of a problem with bacteria and some protists because they don't reproduce sexually, right? So if we're defining a species by the ability to mate and reproduce sexually and produce viable offspring, we can't really apply that definition to bacteria that reproduce through binary fission. Right, or protozoa that reproduce through binary fission. So do you remember what binary fission is? Binary fission means something splits in half, and then the two daughter cells that you get from splitting in half are identical. So binary meaning two, and then fission meaning to split in half. So many bacteria and protists will reproduce, and fungi as well, will reproduce through binary fission. And so how do we define them as being in one species or another? Because they don't actually mate. They don't reproduce sexually. Well, we just do, right? So this is kind of an exception to the rule of the species rule. So we do define species, and we're defining them based on morphology and what DNA they have, what genes they have. We're defining the species that way. But if you have different species that belong to the same genus, then they're related, and then different geni belong to different classes and different families and so on. Okay, but we always, whenever we refer to any living thing on Earth, we always list it according to the binary nomenclature, where we list the genus and then the species name. Right, so I mentioned Lactobacillus earlier, Lactobacillus acidophilus. So Lactobacillus is the genus name, and then acidophilus, Lactobacillus acidophilus, is one of the species within that genus. Right, so there are other bacteria within the genus Lactobacillus. Right. Okay, Plasmodium, by the way, Lactobacillus acidophilus is a bacteria. It's a bacterium, so it's a prokaryote. Okay, below that, notice that we have Plasmodium vivax. Plasmodium vivax is a protist that causes malaria, right? So again, the genus is the Plasmodium genus, and then within the genus, the vivax is one of the species within the Plasmodium genus. But there are other members in the Plasmodium genus. There's Plasmodium falciparum, which also causes malaria, and there's some others. And Giardia lamblia is another protist that causes something called hiker's diarrhea, which we have here in British Columbia. So you have to be careful when you go camping in the woods. You go out for a vacation, a camping vacation, and you see some beavers in a lake. You should not drink the water from the lake where the beavers are living, because it's probably contaminated with Giardia lamblia, which is a disease carried by beavers and other animals that causes diarrhea. And that diarrhea is commonly referred to as hiker's diarrhea, because people go on hikes and then they drink water out of lakes that contain beavers, and they get hiker's diarrhea. All right, so this is the binary nomenclature. Occasionally, because it's such a nuisance to write the whole name, you can abbreviate. It's common practice to sometimes just abbreviate the genus name with the first letter. Sometimes on tests in this course, I'll ask you the name of a particular microbe or something like that, and all I need you to do is give me the first letter and then the species, right? So if you can't think of the genus, it's better if you know the genus, but in the beginning, I'll let you get away with just giving me the first letter. Okay, so this is taxonomy. We have the three common domains: the domain Eukarya, the domain Archaea, and the domain Bacteria. These are the bacteria, the prokaryotes. And then within, we have something called kingdoms. There's a kingdom Animalia. That concept is a little bit outdated. And then we have phyla, which is the next most important way of classifying things. And then within phyla, we have classes, we have orders, we have families, and then we have a genus and a species. Right? The species is sort of the bottom, the most specific. It contains the word specific. It is the most specific way to identify a particular organism. The most specific way to identify an organism is by its species name. Okay, look down here at the bottom of this one. We have Saccharomyces cerevisiae, right? So there are several different fungi within the genus Saccharomyces, and Saccharomyces cerevisiae is the one that we use to ferment beer and wine. Okay, so this table represents a summary of the different types of organisms that we're going to learn in this course. And so first of all, we know about humans. We know what they are. Right? We're also going to learn quite a bit about yeast. So yeast are single-celled fungi, single-celled versions of fungi. These are single-celled eukaryotes. They're a little bit smaller than human cells. They have a nucleus. They usually have 3 or 4 chromosomes, or maybe a few more. They generally produce through binary fission, meaning they produce asexually. Asexually means without sex, but occasionally they do reproduce sexually, and that requires a kind of a genetic change, which is called a mating type switch, which we'll talk about later. All right, fungi are what happens if you take yeast and you put them together into a colony called a mycelium. So when you see mold growing on bread, that is an example of a fungal colony, a fungal colony called a mycelium, or a fungal mycelium. We're going to learn a lot about bacteria, which are small single-celled prokaryotes that do not have a nucleus. They typically have one chromosome only, and that chromosome is circular rather than linear. Most of the other organisms have linear chromosomes. Bacteria have circular chromosomes, which is one of the things that makes them unique. Furthermore, bacteria tend to be quite small. They're usually on the order of 1 micron in size, as opposed to human cells, which are generally about 10 microns in size. And then protozoa are single-celled eukaryotes, which tend to be larger than that. They tend to be 50 or 100 microns in size, and they tend to be very complicated. They actually have internal organs in addition to the nucleus. They might have mitochondria. They sometimes have a mouth. They sometimes have cilia or flagella that they use to swim around. We will also learn about helminths. Helminths are microscopic worms. Microscopic worms. Microscopic worms, the helminths, are multicellular, of course. Many of them are made up of 200 cells or 300 cells. Some of them grow to be enormous sizes. We'll talk about those later. The tapeworms, for instance, are members of the group that we call helminths. The tapeworms can grow to be 10 meters in length. They start out small. They start out microscopically, but if you eat them, they latch onto the inside of your intestine, and they just keep on growing until you have a tapeworm in your intestine. All right. Viruses are not living things. They are a few rogue outlaw genes contained inside a capsule called a capsid. And all they do is they invade a cell and they hijack or usurp the cell's metabolic machinery and use it to make more viruses. And then those viruses come out and they infect more cells, and then those cells are forced into making more viruses. And so viruses just lead to more viruses, but they're never actually considered to be a living thing. Right? And viruses tend to be very small. So if bacteria are considered small because they're 1 micron in size, consider viruses that are typically 10 to 100 nanometers. A nanometer is 1/1000th of a meter. So if you take a meter stick and you divide it by 1,000,000, you have a micron. A micron is about the size of a bacterial cell. If you take a meter and you divide it by 1,000,000,000, you have a nanometer. So a virus is typically 10 to 100 nanometers, something in that range. So if you imagine the Columbia College building as a single cell, the main offices would be the nucleus, and that is typically the size of if you take the main offices and the library together, that would be the size of the nucleus within that cell. If you drove a bus into Columbia College, that would be the size of a mitochondria. So Columbia College could hold 2 or 3 or 4 buses on different levels. And then a virus would be the size of a tennis ball, anything from a tennis ball to a soccer ball. So that's quite small compared to the size of the cell. And then finally, we're not really going to discuss prions in this course, but a prion is a rogue protein. So you may remember a few years ago there was a disease that was circulating around Western Europe called mad cow disease, which is where the brains of cows were basically turning to mush. And that was caused by prions. And so immediately what they had to do in order to get rid of that outbreak of prion disease, mad cow disease, is they had to destroy all the cows that were affected. So if there was a farmer had 1 cow that came down with mad cow disease, they had to kill all the cows, burn the corpses, and make sure no animals or humans ever ate the cow flesh, because it contained these proteins called prions. And interestingly, biologists have always believed that in order to make proteins, you need to have genes. That's generally true, except with the case of prions. So when we really were confused when we started seeing mad cow disease, because we didn't see any foreign genes inside these cows. But after many years of research, they found out that there is a protein that they named a prion, which is capable of infecting one animal and then infecting another animal, and that the protein is capable of reproducing itself without the need for genes. Right? So prions are proteins that are not created on a ribosome the way other proteins are created. They actually build copies of themselves without ribosomes and without genes. And so it's quite extraordinary, but it's bad, right? So we don't want prions around. We have to get rid of them whenever we find them. All right, so these organisms, humans, helminths, protozoa, fungi, and yeast, are all classified as eukaryotes. Bacteria is the only thing that's classified as a prokaryote. And these things are not actually what we would consider to be life forms. They do cause lots of problems, but they're not actually life forms. All right, so remember the divisions of the BC Center for Disease Control? Okay, so the next thing that we're going to do is we're going to basically take a little tour. We're going to take a tour around the world of microbes, and we're going to learn a few examples of each type of microbe that we're going to study over the course of this lecture series. Okay, so before we do that, we're going to move on next to lecture number 2. But before we do that, let's just check and see if we know some of the answers to some of these review questions. Okay, approximately what percentage of microbial species are harmful to humans? 5%, 50%, or 95%? You can pause the recording and then see if you can guess or remember the answer. Okay, the answer is actually 5%. Most of the bacterial species are not harmful to humans, right? Only about 5% of them are. Okay, which of these is classified as a prokaryote? A bacteria, a yeast, bacterium, a yeast, a virus, or a helminth? The answer, of course, is a bacterium. Which of these is not actually a living thing? All right, the answer, of course, is a virus. Which of the following organisms does not have a nucleus? Again, the answer is a bacterium, but also a virus doesn't have a nucleus. Right? A virus does not have a nucleus because it's just a few rogue genes. If this was a multiple choice test, I would ask for only one answer, but because it's it's just the slideshow, there are other. But neither viruses nor bacteria have a nucleus. Whether or not we classify a virus as an organism is a matter of debate. Many scientists don't. Okay, which of the following organisms is multicellular as opposed to unicellular? The answer is the helminths. The microscopic worms are made of many cells instead of just one cell. A bacterium is just one cell. A yeast is one cell. Remember that a fungus sometimes forms a colony. So sometimes we classify a fungal colony as a multicellular organism where all the cells are the same. But nevertheless, it's a multicellular organism. Virus is not an organism, and it's certainly not multicellular. But a microscopic worm, a helminth, is a multicellular organism. What is enology the study of? Is it the study of harmful microbes, the identification of microbes for purposes of oil exploration, or the study of microbes used to ferment wine? The answer is it's used. Enology is the study of winemaking, specifically mainly studying the types of microorganisms that ferment the sugar in grapes and ferment it into ethanol. Okay, in the binary nomenclature, I've done a very bad thing here, I just noticed, and that is that the genus and species name for Plasmodium vivax should always be italicized. And the first one should always have a capital, right? And the second word is lowercase, right? So make sure that you do that correctly. Two words, italicized. The first word has a capital letter. The second word has a lowercase. Which one of them is the genus? So the term, the question is, the name Vivax is the name of what? The genus, the species, or the phylum? Right? Vivax. Okay, and the answer is Vivax is the species name. Plasmodium is the genus name. So in the binary nomenclature, we take both words and we put them in italics. We capitalize we capitalize the genus, and we do not capitalize the species. So make sure you do that correctly. That's important for a biologist to do that correctly. All right, Frederick Banting, famous Canadian scientist. What did he invent? What did he discover? Okay, so he's famous for, in 1921, discovering insulin, which is a treatment for diabetes. It was Canada's first Nobel Prize. Okay, here's a slightly more challenging question. Do you remember what this is called and what it's used for? And the answer is this is a Petri plate, and we use it. The Petri plate is filled with food for bacteria. We streak some individual bacterial cells across the plate using a wire loop, and anywhere that you have an individual colony, if you put that plate into an incubator, which is a warm, humid machine that is warm enough to allow the bacteria to grow optimally and divide quickly. If you put it in the incubator overnight, you come back the next day and each of the individual cells will have grown into an individual colony. And each of these round structures here is a colony. So the bacterial cell gets stuck to the plate and then it divides. So you have 2, then 4, then 8, then 16, then 32, then 64. And by morning, you have a whole colony to look at. So this is a Petri plate, and streaking bacterial cells onto Petri plates is one of the main things that bacteriologists spend their time doing. Okay, that was a very brief introduction to microbiology in general. Let's go to part 2 when you have time. Go to part 2 and then see. We'll take a tour of all the different. I'll give you 1 or 2 examples of every type of organism that we're going to study. We learned that we're going to study bacteria. We're going to study fungi, yeast, helminths, and viruses. I'll give you a couple of examples of each of those different types of organisms. Okay, thank you.